Hello everyone, I'm Dr. BCMR, Assistant Professor at Shastra. Today we'll discuss something, an extension from the experiments of Griffith. What Griffith has found was or discovered is the transforming principle that R strain, Streptococcus pneumoniae, can become S strain, and that is what is the discovery that he has made, but there was no information about the molecule or what exactly is that, what exactly is allowing this R strain to become S strain. So after a decade or two, there are several experiments made. But before going into the experiment itself, let's just discuss about what it could be. The chemical nature of the transforming principle is what we need to find out. So if we want to see it, if the chemical nature of it, let's just see that something, a bacterium is like this of R strain is changing to S strain, transforming into S strain. That must be, then it must be something that is because of some molecule within the bacterium, right? So the bacterium, like any of the living organisms that we know of, are composed of some the biomolecules, lipids, carbohydrates, and proteins, and nucleic acids. So if there is a molecule that is resulting in the transforming principle, it is likely to be one of these molecules. So there were how to determine which of these is the chemical nature of the genetic information is an important question, and then we need to have extensive design of the experiments. So here we have a paper published in around 1944 by Avery, MacLeod, and McCarty. It says studies on the chemical nature of the substance inducing transformation of pneumococcal types. That's a very interesting paper. So although there are many other experiments and there are a lot of technical details about how they isolated the DNA and how they isolated different enzymes that can digest these from different organisms and sera, we are only going to focus on the gist of the experiment. So this is again a nice deductive way of doing things. So what they have done is they have taken the heat-killed S cells, and they did use lipases. They tried several other molecules as well, enzymes as well, to test that lipids and carbohydrates are not involved. And if there is anything that is involved, they have come to, through series of experiments, they have come or narrowed it down to that there are proteins, that is one likely candidate to be genetic information, and there is RNA, and there is DNA. One of these three molecules is likely to be the chemical that is acting as genetic information. So for this, the scientists here isolated or added, first were able to obtain several enzymes, proteinases that digest away the protein, RNases or ribonucleases that digest away DNA, and deoxyribonucleases that digest the DNA but not RNA. Okay, so here we will have no proteins. In this case, we will not have any RNA, and in this case, we will not have any DNA. Let's see it also like this. I will try to use just the first alphabet, P, proteins, RNA, and DNA. I will set up 3 different reactions as depicted in this figure. P, R, RNA, and DNA. I hope you can see that. So in one of the experiments, first experiment that he has done, they have added proteinases. Okay, let's just check in this again, once again, the whole setup. So they have taken the heat-killed S cells, eliminated the lipids and carbohydrates, and they have added one control. So they would take these and add to a flask containing R cells. And after a period of time, they were able to identify that S-type cells are there. Okay, so in this case, they had all the 3 proteins, RNA, and DNA. One of it, I will describe it as control. I will take this as the control. This is test 1 and test 2 and test 3. The reason why I am describing it this way is the important thing about taking from these experiments is about the scientific thinking. We know that the chemical nature of the genetic information is DNA, at least in bacteria, and we don't have to read these experiments except for the way the experimental design was made and how inferences were made. So in the control, what they have done, they have taken the digest, say for example, the heat-killed S strains, and they would have eliminated the lipids and carbohydrates. What is left is protein, RNA, and DNA. And then they were adding the R strain to each of them. R strain is added into the containing these things, except we will get to that again in a while. And then they want to test if there was S strain at all. Did S strain come about, that is because of transformation or not? So what they have done in the first set is just the control, and they were able to see that S strain has formed. In the next one, they have added proteinases in T1. The proteinases will degrade the proteins. So RNA and DNA are present, and they added the R strain, and they wanted to see if S strain has formed or not. It actually formed. So in T2, test 2, they have added ribonuclease or RNases, which would have degraded the RNA. And then they have seen if S strain has formed or not. And indeed, S strain has formed. That means transformation has occurred. And in T3, they have added DNases, because of which DNA would have been eliminated. And there, they did not see S strain formation, or there is transformation of R to S has not occurred, which means that DNA is the limiting factor. If you want to use something like deduction, we would put it this way. We can put it this way, that first thing, in this case, in this observation from control, it shows that yes, R strain can become S strain in the presence of proteins, RNA, and DNA. In T1, the result says R strain can become S strains in the presence of R and D, that is RNA and DNA. In test 2, it says R can become S strain in the presence of proteins and DNA. And T3 says R becomes S strain in the present. It cannot become S strain just by presence of proteins and RNA, which is conclusively telling us in all the cases where transformation occurred, The one common factor is DNA. And in the place where transformation did not occur, the one thing that is missing is DNA. Therefore, the chemical nature of the transforming principle is DNA. That is how they have conclusively concluded that the experiments, that the transforming principle is because of DNA. So of course we have to keep in mind that there are other aspects as well, such as this DNA is the genetic material in majority of the organisms. You can take plants, animals, or fungi, bacteria, archaea, you can take any of them. But in viruses, the viruses are made up of proteins and some genetic information. Some viruses have DNA as their genetic material, and some viruses have RNA as the genetic material. If we were to consider all the propagating entities, if we can take about viroids, which can also propagate, which are acellular. They do not have anything else other than just RNA. So RNA can act as genetic material as well. So what Avery, MacLeod, and McCarty have actually found is very important from the biochemistry point of view and also from the biology point of view. That all of the genetic information in most of the living organisms that we know now is made up of DNA. And then started the struggle or finding the structure of DNA. How does a chemical carry information? That is something important and profound at the same time. I hope you enjoyed the class. Thank you very much.